Well, thanks for that introduction. Yeah. So my name's Zach. I'm a postdoc or research officer in the Brain Cancer Lab, and I'm here to talk to you about sort of my journey, my scientific career, and what I'm working on now. So my journey to get here, what's really been driving me? My scientific career has really been defined by my parents. In particular, it's really been defined by my dad. So he doesn't have brain cancer, but he does have an acquired brain injury. So I certainly really understand what it's like to see someone you love undergo this horrendous incident, deal with these neurological effects, making accommodations, and really dealing and navigating what can be a really confusing health system. So I grew up in Ballarat, finished high school, came to University of Melbourne, where I did my undergrad, and I really started to get an interest in the brain. And I really, as I said in my video, I really like the brain. I think it's fascinating. And it actually ended up with me doing a PhD in Alzheimer's disease. And here I am at my graduation with a very proud mum. And during that time, this incident with Dad happened, and it's really just cemented myself in what I do and why I do it, acting within the brain sphere. So I finished my PhD and I started to move out of Alzheimer's disease, and I've actually made the jump. Physically, not so much, because this is actually an arrow from my old lab to my new lab, but certainly scientifically a little bit. But what I've really brought with me is, a nice skill set, the background knowledge to be able to tackle, these issues within a brain cancer context, a real new set of fresh eyes, which I really think I can bring to the table. And it's certainly a real privilege to be able to come and work here as an early career researcher, being supported by this fellowship. It's a real privilege to be acknowledged and listened to and heard, and it's a really progressive way of how we do science, and I'm truly honoured. And that really brings me to my project and what I'm currently working on. So I'm working on glioblastoma, or GBM, which I think we all know is the most common and deadliest form of brain cancer. And I think we can all agree that it's not just a single problem in isolation, it's actually a real collection of problems. But to me, one of the biggest problems that really stands out is that lack of drugs and therapies that we have to help manage disease. And I think that's really exemplified by this table here, which is a list of everything that's ever been approved. And it's short and it's really not good enough. So leveraging my skills, new technologies, and our connections, my connections within the Brain Cancer Centre, I'm really looking to address this. And so when we try to think of why there are so few drugs that we have, it's really important to recognise what the current drug discovery pipeline looks like. And here it is demonstrated. We can see we have sort of four main steps going from R&D all the way to review and approval. And it's long and it's expensive, 10 to 17 years, average cost $2 to $3 billion. But now I'd like to introduce this alternative pipeline when it comes to different to this traditional model, and it's called drug repurposing. So what it is basically is taking an old drug and putting it in a new setting. And so there's really two major advantages to this. The first, of course, significantly cheaper, but the one that we're really concerned about is time. Significantly shorter time to market means quicker outcomes for those with glioblastoma, and we really know that those with glioblastoma, time is scarce. So what does this look like? Well, part of the project I'm working on is looking at new treatment targets within glioblastoma. And so through various different approaches, I aim to find new genes and proteins that I think look promising to target. So, for example, I might find a protein that I've represented here in green, and I think it contributes to glioblastoma, and I want to target it. And then through my literature search, I find this same protein has actually been implicated in a separate disease setting. And importantly, there's already been a compound that's been developed against it, and importantly, it's safe and effective. And that makes it the perfect candidate for drug repurposing, taking this old drug, putting it in the setting of glioblastoma. So the question most often asked when this concept's brought up is that, does this actually work? Has it worked in the past? And the answer is yes. And there's several examples, but I think this is the best one. So this is a scan of a paper published in 1996 from a group at Pfizer. And all you need to really focus on is that they were interested in this protein called 5-cGMP phosphodiesterase. And this protein's involved in regulating our blood pressure. And they thought, well, if we can develop targets against this, we can use drugs in a number of different sort of heart disease settings. And so they developed a number of drugs, and they found one of them called sildenafil here. And they took it to clinical trial, and they said, okay, let's see if it works. And really what they found is that it actually had a modest effect at regulating blood pressure, But surprisingly, it was actually a whole lot better at something else. So I think this is a really good example of one of the biggest pharmaceutical companies in the world with the most considered and expertise drug development team has developed a drug best in class so far beyond their original intentions. And so that's a nice serendipitous discovery, example by chance. So how does it look for me? So as I mentioned, part of my project is looking for new treatments within glioblastoma. And I've identified over 100 targets that I think look really promising to modulate. But then the issue is, well, what drugs do I choose to modulate these? And really there's just about over 5,000 that we could potentially choose from. And these include drugs that are already approved, that have high quality data, and ones that we can purchase from our commercial suppliers. So what I do is I use this really computer-driven approach to really narrow down and focus on particular combinations that I think are going to work. And so with this approach, I aim to look at 100 drugs to test. And then, of course, well, the next step is how do I test them? What do I test them on? And this is where I'd like to introduce this concept of neurospheres. So these are really our most precious and valuable samples in the lab, and we're truly grateful and honoured to use them. So it's been a really critical part of my project in establishing these. So from those who undergo surgery at Royal Melbourne and choose to generously donate their tissue, I can process it, put it into a dish where it will continue to grow and grow and grow. And this is what they look like under a microscope. We call them neurospheres because obviously the neuro part refers to them being from the brain, but spheres because they actually grow out in this sort of ball-like shape, sort of floating around. And the really important part of why we use this model is that we've been able to show that when we grow and maintain these spheres like this, they actually better mimic the tumour as it was in the brain. And the better they mimic the initial tumour, the more confident we can be with our potential drugs in knowing that they work, and the better chance we have of taking them onto the clinic. So when we treat our neurospheres, we want to see how they respond to drugs. And one of the ways we do that is by taking images over time, and that really helps us understand the dynamics of how these drugs cause their effects. And you can see in this video how my treated cells, they'll eventually die and burst, while my untreated cells, they'll just keep growing and growing and growing. So we want to look at lots and lots of individual drugs, but we actually don't think that a single drug is going to be the cure-all for glioblastoma. We think it's actually going to be a combination of individual drugs. So how do we look for combinations? How do we get this really multi-pronged attack on different levels? And we really use this elegant approach. So when we treat our neurospheres with our lots of individual drugs, we use those images as well in our analysis pipeline. But we also do a lot of other measurements, thousands of different parameters, see what goes up, what goes down, what stays the same. And basically what we do is we sort of bring them all together and we form a drug signature, so a signal signature. And then, again, using this really nice computer-driven approach, we look for complementary signatures. So if they overlap, are they going to work together? So you can imagine if I find a drug that can maybe kill 75% of my cells in a dish, then I can look at these signatures and think, well, actually, I think this other one's going to get it over the line. And the really nice part of this is that we don't have to, test every single possible combination. We can really focus on what we think is going to work and make sure that it actually does. And here's what it actually looks like, but can potentially look like. We can see that this, drug in yellow, eventually it will cause my cells to die, but the combination of the yellow and the blue gets that drug to kill these cells a whole lot faster. So, in a nutshell, we bring it back together. My project is looking at generating this list of drugs, testing my neurosphere models, and do these really detailed analyses. And right now it's very exciting because I've established my models, I've established how I'm going to analyse them, and right now I'm just in the midst of finalising my list of drugs to test, and then we're ready to hit the ground running. But the even more exciting part is this, because we don't want it to stop just there. Being based within the Brain Cancer Centre and WEHI, I work with individuals who have the skills to take what I find and move them directly into the clinic. And, that's a truly unique place to be in, and really not seen elsewhere around the Parkville precinct, I believe. We have the opportunity to provide real-world change and make genuine differences to those with glioblastoma, fix this issue of our lack of drugs, and that's why I'm here. So with that, I'd like to make some acknowledgements. Alongside my own personal experience, our consumer team is really critical to what we do. I have absolutely benefited from their input, and I think I speak for the lab when I say they have as well. They're instrumental in shaping our research from the get-go. And finally, acknowledgements to the lab and the others that have been involved to make this happen. It's a fantastic place to be in and a great place to come to work to every morning. So to our consumers, patients, individuals who have donated their tumours, all of you here today and online, just want to say thank you, and thank you for giving me the opportunity to present my research to you all.